As we have seen when we talked about the emergence of multicellular animals, a very important mechanism of genome evolution is the expansion of gene families, groups of genes rather similar to each other. Gene families are quite frequent in today's genomes. They are the result of duplications of entire genes, and what is really interesting is that each member of the family can specialize in performing a particular function or working in a different cell type. Generally speaking, when a whole gene is duplicated, usually together with its regulatory elements upstream, there are several possible outcomes. The first and simplest, especially in the absence of clear evolutionary advantage for the gene and its protein, is that the new copy, or the original one, is inactivated due to some mutation, so it becomes an inactive pseudogene and nothing happens. Another possibility is that the new copy retains the original function, so there is an increase in gene dosage and in the amount of protein synthesized. This has happened, for instance, recently in human evolution in the number of copies of the gene that encodes amylase, an enzyme that we need in order to be able to use starch as a source of energy. If the evolutionary pressure to increase that protein is strong enough, as in the case of amylase, then we will see how the number of copies of this gene increases in the genomes of that species. But finally, and this is the most interesting part, at least to me, if the original copy retains its function, then the new copy can be used to experiment and acquire some changes that will lead to a new function or will change somehow the way in which that gene works. This process is called neo-functionalization, when the new copy takes on a new functional role, or sub-functionalization, when the gene does pretty much the same thing, but there are changes in the regulatory elements that control its expression. So each copy will be active in some particular tissue type, for instance. A good example, a classical example of this process is afforded by the genes encoding the globins. Globins comprise a family of genes, a family of proteins, present in different versions in all kinds of animals. When we compare modern globins, we see a picture more or less like this one, with a hypothetical ancestral globin about 800 million years ago, that is before the arrival of animals, which duplicated to give modern neuroglobin on the one hand and all the other globins on the other. Then about 550 million years ago, during the Cambrian explosion, there was another duplication which originated modern cytoglobin and myoglobin, and a later duplication about 450 million years ago that led to alpha and beta globins. These, the alpha and beta globins, are actually gene clusters, because each of them underwent subsequent duplication events, so that we have several alpha globins and several beta globins. It is very important to understand that these copies in the same species, myoglobin, and alpha globin and beta globin, for instance, are not the result of speciation, but of duplication. So all these globin genes in one species are known as paralogs because they are the result of duplication and divergence, whereas the versions of one of these globins, let's say alpha-1 globin, for instance, in different species, imagine mouse and human, are known as orthologs. These are the result of speciation. So gene duplication has been one of the main forces at work during genome evolution, creating gene families that can perform a wide variety of molecular tricks, enabling organisms to become more and more complex and sophisticated. And there is a final type of duplication that seems almost impossible or extremely unlikely, but in fact has happened several times over the last 500 million years, and that is the duplication of entire genomes.